Hi, hello to everyone. My name is Nicoletta Contaldo. I'm a research professor at the University of Bologna, Italy. I've been working on phytoplasma diseases for more than 10 years, and my main research topic is related to their epidemiology and to the study of their biological and molecular features. Why Asteriellos-related phytoplasmas occur on Grapevine on South African regions, the situation in Italy is quite different. But generally speaking, as you can see here from the world map, grapevine yellows are associated to different phytoplasmas in the different geographic areas where grapevines are cultivated. The main yellow species in grapevine in Europe are known as Flavescens-Doré, FT, phytoplasmas belonging to group 16S are 5C and 5D, and Bois Bien, Candidatus phytoplasmas solani, belonging to the group 16S are 12A, and they are transmitted by Scaphideus pol and Ialetus obsoletus ignoré, respectively. The symptoms on the leaves are yellowing in the white grape and reddening on the red ones, backward curling and shortened internodes. And on grape we have flower abortion and shriveling of the berries, and on the canes lack or lignification. But the same symptoms that you can observe on the plant are associated to different phytoplasmas. As you can see here, these are the different restriction profiles obtained by from different phytoplasmas, FTD, FTC, and BN, that are clearly distinguishable by RFLP analysis and by after digestion with the restriction enzymes. And this is the general methodological scheme used for the identification and classification of phytoplasmas on 16S rDNA gene. So we start with DNA extraction from leaf midribs and doing a direct and nested PCR with universal primers, and after checking the positivity of the samples in agarose gel, we can use two different options, RFLP and or sequencing, for the identification of the phytoplasmas amplified. And now I want to show the results of 5 years monitoring of grapevine yellows in Italy. As I said before, Italy, the Italian situation is very different from the South African one, because the grapevine yellows in Italy is puzzling, because the different phytoplasmas are identified in different regions. Here you have the location of, mainly the location of Flavescens-Doré presence, and as you can see is restricted to the northern part of the country, while the Bois was identified in samples collected in the central and the southern part of the country. Moreover, a growing number of diverse phytoplasmas have been found in Italian vineyards, together with the historical Bois and Flavescens-Doré phytoplasmas. And as you can see here from the pictures, no difference in symptoms from the BN on the left in Lambrusco and the FTD in Pignoletto from 16S10B in Cabernet and Asteriello 16S1B in Chardonnay. So we focus our study on the ecological variables influencing the grapevine yellows epidemics, and we analyze grapevines, insect factors, and phytoplasmas epidemiology. So here are just some examples of experimental design. You can see the position of insects, grapevine, and spontaneous plants collected in and around the vineyard. All the samples were subjected to molecular analysis. For the spontaneous vegetation, here are just a few examples of plants or collected plants, mainly asymptomatic ones. And here there are the major vectors, insect vectors collected in and around the vineyards. Around the vineyards and the polyphagous insects, the main polyphagous insects collected, identified and collected were Ialetus obsoletus, the vector of Bois, Orientus ischide, vector of Flavescens-Doré, and a protective Fusco venosus phytoplasma vector. And the Parophagus insect, Scaphideus ball, vector of Erasmus and the Erasmoneura vulnerata. And there are just some of the results, the molecular results that we obtained from the different insect species collected in the different provinces. And here you can see that how many phytoplasmas, diverse phytoplasmas we could identify. Not only 16S12A, the BN, and 16S5C, the FT, but also the aster yellow, the 16S1B, and the group 10, the group 6, etc. So what we can conclude, that the presence of the diverse phytoplasmas in grapevine showing yellow symptoms is indicating the possible presence of diverse phytoplasma sources in and around these vineyards. So considering the age of the majority of the vineyards monitored, it can be speculated that the environment plays a key role in the presence of the diverse phytoplasmas, making it difficult to manage the disease spreading. Because while the FD and BN phytoplasmas have been very well studied since their first outbreaks, little is known about epidemiology or the other phytoplasmas detected. So the results of the survey indicates the need for more accurate surveys to verify the presence of other phytoplasmas and their potential epidemical role in the yellow disease of grape. Thank you very much for your attention.